Hi, everybody. Welcome to New Frontiers in Functional Medicine, where we are interviewing the best minds in functional medicine. And today I'm talking with one of the best. You can see her here if you're watching us on YouTube. If not, let me tell you about her, the one and only Dr. Betsy Redmond. She has been a friend and colleague of mine for my entire career, really. We were together at Metabolics Laboratory many years ago. So today, Dr. Betsy Redmond is a senior education specialist at Diagnostic Solutions Laboratory, and she's also a nutrition consultant. She is conventionally trained. She's got a master's degree in clinical nutrition from Emory and has a doctorate in nutrition from the University of Georgia. She has over 15 years experience in functional laboratory research and education. She was part of our brain trust at Metabolics. In fact, we all leaned on her brilliance. Welcome, Betsy, to New Frontiers.
Oh, you're going too far.
It's always great to be able to hang out with you. I'm very excited. So, I'm just thinking about Jeff Bland. He started the Personalized Lifestyle Medicine Institute, sort of the next generation of what we're thinking about, where we're headed in functional medicine. And I have been to many of his conferences, and we all wait with bated breath for metabolomics panels, for these, more robust panels combining organic acids, amino acids, fatty acids, like just bringing that knowledge forward. So we started back at Metametrics, you and I and the whole team over there, looking at smaller panels. Like it was the forerunner of being in the omics era. But I'm very excited to be talking to you today about, the metabolomics that DSL has gotten into, and bravo you guys for kind of really taking the bull by the horns and bringing this to functional medicine. So tell me how you got there, how you guys decided to offer this, and what you're doing, and then we'll talk about some of the specific markers.
Okay. Yeah. We decided we wanted to offer, organic acids, amino acids. It's, in the functional space. And, but we wanted to be what's current. Mm-hmm. So, we looked at it, and organic acids used to be more like, this is high, it might mean this, or you might need to take this supplement. But metabolomics really looks at a bigger picture. And metabolomics really started with maybe just small molecules, but they've really pulled out. And so you can see they'll look at things like aging or metabolic concerns. They'll look at a group of people who have something and a group of people who don't. And then they'll look and see what's the difference. And oftentimes when they make these charts with all the bubbles, it'll be like tryptophan pathway, not necessarily specific markers, or the phenylalanine pathway. So we wanted to look at it in a whole pathway. Mm-hmm. And so that's why we wanted to put them together. So if you look at our report, it will have, branch chain amino acids right with their alpha keto acids, or tryptophan, and the whole pathway. So if you look at a pathway next to it, you can then start to see where the issues may be.
Right. And we will link to a sample report on the show notes, people. So access that. In fact, you may want to pause this and just take a second and grab it, because we're going to refer to it. I got my test done. Betsy and I were going to record this a few weeks ago, and then we stopped midstream, and I said, I might as well get this done so that we can talk about it. So on one hand, back in the day, we were looking at organic acids. If high, give B6, if high, maybe you have some yeast or something. We had more simplistic explanations. We were in the research as much as we possibly could be. I know we put tons of energy, but there just wasn't. But now we're really, truly entering the omics era, to your point. And now we're able to look at, many more compounds and kind of really dance around what's happening metabolically. And also, to your point, what's very exciting about this is we're looking at disease associations with imbalances here. We're looking at what healthy cohorts look like. We're looking at what aging looks like. So I think it's pretty exciting stuff. I mean, this has been
I think it's great.
A passion project for you.
It has. And I was going to say, there's a lot of markers, especially if you're new, because we also wanted to, like, if conventional clinicians want to see what is that, so I want it to be easy to look. if you go to you can take any of the markers we have, or really any organic acid, fatty acid, amino acids, and look up HMDB. So the Human Metabolome Database. It's
And we'll link to that on the show notes also.
Mhm.
Do you happen to know the website off the top?
You know
of your
what?
head?
When I want to do
We'll link
something
it.
like if I wanted to look up citric acid, I would just go to like Google or a search engine, put in citric acid HMDB. It'll come
It'll
right
take you
up.
right there. Okay.
And so it gives you what's currently known. I mean, some of the things they don't go in maybe as deep as we do. And then they'll also give you all the synonyms, which is really handy because sometimes things are called.
That's exactly right.
different.
That's right. That's right. That's right. So you go on Wikipedia and you can see one name, and then on the Human Metabolome database, actually they'll corral them all together.
Yeah, they just have synonyms.
Wikipedia does that not so
yeah,
bad.
and they're very, sometimes Wikipedia doesn't go as deep to maybe as obscure compounds. But
I want to just say too, I'm looking at my report, and I've given you permission to talk freely about it. But you've got and if you pull up the sample report, you'll see that there's a down and dirty reference. So if you don't want to geek out on metabolomics data, and if you don't want to go to the Human Metabolome database, which is where Betsy lives when she's not in Georgia, you can get it down and dirty. This is where you need support. You need support in toxins. You need support in, certain amino acids. We'll talk about the aging, because that's, an interest area for me, not just because I'm getting older, but because, so for clinicians who are working in a really busy practice and you don't have time to do the drill down, there's a easy, ready reference, front page summary cover that will give you really clear direction. Let's talk. So let's talk about. And actually, let me just say, giving you clear tell me the clear direction that you can get. What can a clinician glean from this first page that's going to guide, from the report as a whole, that's going to guide what they're doing with their patients?
Right. It goes in like, I always got to use hand motion. So it goes in like 3 steps. Like the first one, the top of the front page is really broad. So it's going to look at a big overview. And then the bottom of that is kind of a further description. So we have 6 categories that we're looking at, like energy, metabolism, and then we look at all these amino acid breakdowns, nutrition, toxins. Then we look at the microbial metabolite stuff. So we're looking at 6 different areas, and we look at all the markers in there, and then we just kind of add up how many of those are outside the reference range. So we give a score. So the front page is based on the 95th percentile, because that's by labs what they're going to put as out of range. So that summary score of each category gets put at the top. But then if you look at the bottom half of the front page, it breaks it down. Like, is it glycolysis? Is it the Krebs cycle? Is it the phenylalanine pathway? So it kind of gives you an idea. And then the last layer is to just go through the test, and it'll tell you specifically, because we're doing it based on the 95th, because that's proper lab work. But if you go through, you may find other ones that are creeping up that meet the story that you want to tell.
Yeah, so then you can move into the heart of the laboratory data itself, and you have, which I absolutely love, coming from our lab background, you've taken the time to divide them into quintile reference ranges. So we can see the outliers, the 95th reference interval, so the frank highs, the frank lows, but then we can also see where we fall within the population as a whole. Am I right in the middle? Am I a little bit high? Am I trending down? And then as clinicians, we get to drive the car much more efficiently,
Right.
I think, and make good, strong clinical decision based on trends and not just frank disease. Like, we're moving. One of the things that Peter Attia said that I think is very apt, it's easy, is he talks about the current medicine model being sort of medicine 2.0. Don't come to me until you have a disease,
Right.
Then we'll diagnose the disease. But we know that pathological changes happen earlier, way earlier, maybe decades earlier, maybe in utero even, and it's only with these subtle, intervals, it's only with being able to look at the data much more clearly can we can we perceive those changes, early on and actually make a difference? So
yeah, I mean, I think that's everybody with IBS, isn't it? Going
Yeah.
to the conventional care and they're like, oh, we did a colonoscopy. You don't have cancer. Thanks.
Yeah,
Yeah.
that's right. That's right.
So-
Yeah. And you haven't pooped in 3 days, no big deal. Okay.
Come
That's
back
not
when it's 10.
constipation. Yeah, that's right. So let's talk about some of the aging analytes in here. Longevity has moved into the front and center of our conversation, maybe because those of us who've been around functional medicine for a while, we're all marching into our 40s, 50s, and 60s. And we care about it. But I think we're getting more and more in our space that the aging phenomena, the changes that happen to the body in the aging journey, actually drive the diseases that we're trying to keep at bay in functional medicine. So how can we assess that?
Well, I mean, it's hard to study chronic disease and not also study aging. I mean, they kind of all move along together. Yeah. When you look at some of those research studies, I think, I've always got a stack of articles, but, so this
yes,
one
you
is
do.
by Johnson. It's and it'll be, I'm assuming, all in the show notes, but they have a really great graph. And it has chronological age on one side, biological age on the other, and then it kind of, how much they correlate. So it has an R-squared value of like 69.69. So about 70% of chronological, biological age is accounted for with chronological age. But there's such a huge variation. And I think that's where we want to kind of
That's where it gets interesting. Yeah.
Yeah, like they divide it by men and women, but there's like, two men, one, that are both 20. One looks like he's like 15 and one looks like he's 45. There's a 70-year-old woman who looks like she's 40 and another one who looks like they're 95. And what they also found was that as people age, like they tested them 5 to 10 years later, the people who were aging faster were still aging faster.
In this article, which is extremely cool, not a whole lot correlates. Not a lot of these biomarkers correlate with the chronological age journey, but certainly some of them do. But the biological age journey seems kind of rich here, things that we can actually action and make different. But so talk to me about some of these markers that are really interesting to you and why. And then if you want to, let's talk about some of the markers you found on my test. So talk to me about this paper, what's interesting there, and then let's talk about the test and what you guys have and what we can look at as clinicians.
Yeah, I mean, so I was saying that, they were looking at markers related to biological age, and one of those was homocitrulline. So, homocitrulline, when it's higher, it was higher in people who had higher biological age.
Got it. Okay. All right.
So
and that
so it comes off of the urea cycle. So it's also, related to like really high protein intake, any kind of kidney, impairment function. So you can see higher levels. And there are articles talking about, protein carbamylation, because that's what it's called. When you make homocitrulline, it's carbamylation, and it's been associated with aging.
So it's a change to the protein structure.
Mm-hmm. Yeah. So just like proteins can undergo alterations with glucose. So
yeah,
you can make, so the same type of thing, but it's with the isocyanic acid that comes off of it comes off when you make urea. It also comes off, and you can also get it from cigarette smoke. It binds with the lysine residue, and then you make homocitrulline.
So is it a surrogate marker of some sort of toxic process, or is homocitrulline itself toxic? Do you know that? Just out of curiosity?
It's
kind of interesting because it's a byproduct. It's from smoking, can cause this structural change to protein, and aging, we know causes the structural change to many proteins. And this is, one of those markers. That's so fascinating.
So I will say yours was low.
Woohoo! That's awesome. That's well, and how if we've got somebody with a high homocitrulline, what do we do about it?
Well, I mean, it's going to be related to urea production. So I would, well, first, if you're smoking, I would
Kidney
stop
function.
that, look
Right.
at kidney function overall, like,
Yeah.
and on our test, you could look at, creatinine, albumin, look at some of those things. When you go to make homocitrulline also, it also shoots over and makes orotic acid. So more orotate would be made. So if they're both elevated, I would, is it somebody who's on a high protein diet who maybe needs to pull back a little bit? Maybe they don't have the full function for that.
Yeah, that is so fascinating. So so there's been this trend towards massive consumption of protein in our space. We blogged on it recently. Actually, we did a bit of a dive on it, but sort of there's a movement towards consuming, up to 2 grams per kilogram body weight, or a pound body weight. So high protein intake. And it makes sense to me that it wouldn't be ideal for all of us. We're all different. And I know there's compounds that we make in the gastrointestinal tract from protein or from certain amino acids that aren't necessarily our friends. So if we're consuming a lot of protein and we have dysbiosis, we might see some of those jack up and be associated with insulin, like classically, excuse me, not insulin, with inflammation, like classically indican.
Yeah. So I think that's one of the things I think you're right. Everybody isn't going to be the same. So maybe you can't handle that.
Right. Or at least not with your current, metabolic snapshot. Right.
So, and so everybody's going to be a little bit different. So it might help you tailor that.
Interesting. So homocitrulline would be one of those compounds, both if it's elevated, we're thinking about accelerated aging. Specifically, we're thinking about kidney function. We might see it in somebody with higher protein intake who is not doing as good of a job with it.
Right.
What are some of the other markers associated
overall with yeah, I mean, I think that there's a good article, and because I'm all about the articles. Because I think there's so many issues though. So you can start looking and, what are they doing? Are they looking at people who did age well, who didn't age? I think it just depends on what you're looking at. So you don't want to automatically read a study and say, well, that's what I want to do. Just because older, centurions might have that, but is that what you wanted? So there's that study where they look at people who are older in China, like who live in a town that there are lots of centurions, and they compare it to a regular town where there's not lots of centurions. And then they found some things, like they were more likely to have higher tyrosine, higher citric acid, higher lactic acid, and higher valine. But so, yeah, and I think the citric acid, that's also so related to diet. So, more plant-based diet tends to have higher citric acid. It can also go up if there's a issue with renal function, but your citric acid was good.
I'm happy to hear that, especially because just recently my brother went through kidney stones, and that's, classically associated with low citric acid, and dehydration. Citric acid is one of the interventions, lemon water. We always recommend our patients to drink, and very seriously. I mean, it can absolutely make a difference in the formation of the calcium oxalate stones. So citric acid is evidence of a good diet, and it's seen in people who are living long lives. Are they healthy? Did they look at that group and define that?
They didn't really define that so much, but you see it in other studies where they look at people on more plant-based diets, higher polyphenol, citric acid.
And it's associated with health?
Yeah. So I think also we say, like, don't eat a bunch of pectin right before you take the test. We have a list of some food because that can shoot all those up. The other, one of the other things that we're looking at in aging is a tryptophan, decreased tryptophan. And so that pathway, I'm sure, is fascinating because it does lots. So that's one of the ones that you had that surprised me, was a lower tryptophan.
And that's it. So, and that's not a great thing. I know it's kind of, it's
I'm not gonna,
I
well,
eat plenty of protein, but I have struggled with like my albumin, which in circulation is, that's another longevity metabolite. And mine tends to be a little bit on the lower side. I think I actually may be somebody who needs to stay on eating decent protein. We'll see. I mean, that might be a piece of the puzzle, but my tryptophan was low too. That was interesting to me.
I mean, overall, you looked great. I just want to be
good. I look youthful.
Yeah, look like a 16-year-old.
So tryptophan, I want to understand about tryptophan, but what made you determine in general, that I had a metabolomic pattern of somebody younger? So first tryptophan, and then give me your
Yeah, you know what?
analysis.
I'll give you the rundown, and then we can go through the whole tryptophan thing. So you had higher citric acid, you had higher alpha KG, you had
alpha ketoglutarate.
Yeah, you had branch chain amino acids that were not high. So you want to have those in the sweet spot. You had lower quinolinic acid, your methionine was lower, and your taurine was good. So those things. The two things that I noticed was you had a slightly elevated pyroglutamic acid, which might be a glutathione thing, and,
yes,
and then lower tryptophan. But you had kind of a slightly different picture. So tryptophan can go on several pathways. So tryptophan, can go over to make serotonin. Everybody's pretty familiar with that. And then, so the breakdown product of that's going to be 5-hydroxy-indole acetic acid. And yours was good. So you seem to be making that. But why normally when I see a low tryptophan, so tryptophan also goes to kynurenine and then to make indoles in the gut. So the kynurenine pathway, normally tryptophan will throw out some, the TDO enzyme in the liver. It's making some kynurenine. That kynurenine pathway goes through a lot of steps. It makes quinolinic acid, that makes NAD. Like that's what it's churning out. But if there's any inflammation, it switches over into that extrahepatic IDO enzyme. And so if there's, TNF alpha, if there's IL-6, different inflammatory markers, it just pumps up that IDO enzyme, and that pulls tryptophan down to kynurenine pathway.
And so we'll see evidence of inflammation. That's the pattern you usually see. So when you see a low tryptophan, you're flipping the page to look at inflammation.
Well, I'm looking on the pathway because we also report kynurenine itself, like not kynureninic acid, but kynurenine, that first step. So you can look at the kynurenine to tryptophan ratio, the KT ratio. And that's been associated with chronic disease. It's been associated with aging. It's considered
so a high K and a low T.
Right, the other low T. But so it, you're going to be pulling that down. So normally we see, it get pulled down, and when you pull down so much of it, there's so much inflammation. Sometimes people will, essentially run out of tryptophan to make serotonin, and they might be associated with depression. So yours didn't really have that pattern, but looking for inflammation, why is the tryptophan getting pulled down that way?
And inflammation, of course, there is, unless we're very actively working against it, the volume on inflammation is going to turn up on the aging journey. So I can work on increasing my tryptophan with tryptophan foods and, my gut microbiome, but I'm glad to hear that inflammation is a non-issue via the kynurenine pathway. That's awesome. Can you tell can you infer from this that I'm making adequate NAD from this information? Because that's another sort of a hot compound that we're thinking a lot about.
So what you see in research, what I've read is looking at that pathway will, you'll end up making, more kynurenine. You'll have in aging a higher KT ratio. But then it goes down the pathway, but you have elevated quinolinic acid. So, elevated because you're not going all the way. So quinolinic acid has a specific enzyme, the QPRT, that it's very sensitive to inflammation. So then you end up pushing down the pathway, you end up with higher KT, higher quinolinic acid, but you're not going to make more, NAD.
Nice. Mhm.
Yeah, so you end up with low NAD. So you're not making it because inflammation is stopping the quinolinic acid, and then you end up with higher quinolinic acid. So you want to check out all the spots of that pathway. And then, the pathway is really heavily dependent on B6. So that's also something to look at. Probably, there's some good studies looking at mathematical modeling and when B6 is going to come up. So trying to look at it, I mean, we've always looked at it based on tryptophan load studies, but most people aren't doing a tryptophan load. But in the mathematical modeling, they find xanthurenic acid can become elevated first, even though there's lots of B6, enzymes that require B6 in the pathways, and xanthurenic acid generally comes up first. Yours was a little bit elevated.
It was, yeah, I'm looking at it now. So I'm going to say that I don't have a lot of inflammation. So the kynurenine pathway probably is going on to make NAD, but I don't have that. There's not a whole lot of action here because I don't have that much tryptophan. So I could probably use some tryptophan to support NAD synthesis. And then to your point, my xanthurenic acid's a little bit high, and so I could use some B6, and that could help in driving tryptophan down to NAD.
Yeah, I mean, I think it's always good to make sure you support that whole pathway.
Yeah.
I mean, there was a study, and I just always thought it was so interesting. They looked at, there was people who lost weight, and as they checked their actual B6 blood level, PLP, and then they had them lose weight, and they had less inflammation. And so with that, they weren't chugging through this pathway, but their actual B6 blood level went up. And they were saying, well, probably because they didn't need so much B6 to chug through this inflammatory pathway, because pretty much everything requires B6.
Right. So by losing weight, they in effect turned the volume down on some of the metabolic damage that was happening, turned inflammation down and preserved B6 status. Is that what they were suggesting?
Yeah.
That's pretty cool. Yeah, that's really interesting. All right, what else did you want to talk about on this, on some of the markers associated with aging? And then I want to spend some time talking about why my pyroglutamate might have been up, because you had a really compelling reason that made sense in the context of my life. So other markers, and then we'll move on to
well, and the pyroglutamate, I was, one of the markers that goes down with aging is glutathione.
Right. So you must see pyroglutamate. So pyroglutamate indicates the glutathione pathway is sort of being taxed. It's being used, it's being taxed, it needs support. Is pyroglutamate in the literature? Does it rise when glutathione, like in aging?
I don't know if it specifically rises in aging. They talk about glutathione itself. So when pyroglutamic acid goes up, it can identify that you have inadequate amounts of sulfur amino acids like cysteine, or you need some additional glycine. So if you think, you need cysteine, glutamic acid, and glycine to make glutathione.
And so we want to be thinking about that with a rise in pyroglutamate.
Right.
All of them. We just want to think about glutathione, but all of those individuals. I want to just say that pyroglutamate has cool research on, rising with Tylenol toxicity.
Oh yeah, that's its big. I think that's its claim to fame.
It's fame. Yeah, I mean,
that's where they realized. It's a hepatically induced. Yeah,
because that's Tylenol steals glutathione pretty efficiently.
I think there's that old classic article about the young doctor who, somebody has acetaminophen poison toxicity, and he gives them a bunch of glycine and stuff.
And NAC. NAC is standard of care. IV NAC.
Yeah. So when you look at that, so when you go to your methionine pathway, your methionine's lower, which is great, because you see that in research, like, every, all sorts of animals in research have lower methionine. Methionine restrictions, a big thing. I don't think, it's obviously not a solved issue. It's sometimes methionine restrictions a little easier than caloric restriction, but in some of those research studies.
Well, do you know why is an elevated methionine associated with aging? Do you know? I mean, that's like a classic amino acid in meat again, right? Maybe would it be high in
Yeah.
would it be another thing that we want to maybe nuance, like we just don't necessarily want to be consuming, 3 steaks a day. If we're paying attention to the literature on methionine, but
Right. Yeah. I
do
mean,
you
I
know
would,
why we want it lower? Like, what do they say? What do they speculate about in the literature?
I mean, I think that there's, we see it higher, the first thing I think of is impaired methylation, just because
Okay.
it's
Yeah,
involved in methylation.
sure.
And that's not something
Yeah.
like, oh, methylation's great, let's turn up the volume all the way. So you kind of want to pull back from that. And then, so yours was a little bit lower, which was good. I'm always looking for a balance. I don't want really high, really low.
Yeah.
You had some of the markers after it, cystathionine, that were higher. And some of those are, cystathionine itself can become elevated if there's a need for B6, B12, folate, those kind of markers can impact it. Because
Yeah.
that's,
And I do think I need support in methylation, even as people know me, researching specifically DNA methylation, but I need to tend to my methylation cycle. I mean, my homocysteine will definitely trend up if I'm not being mindful about consuming adequate methyl donors, for sure. So that does make sense to me. I want to actually, going to your point of high methionine, in negatively influencing the methylation cycle, that really famous Jill James study. It's looking at the methylation cycle in individuals with autism. It's a super old study, but it was impactful on me. It was in 2004. And so methionine was high in this population. Homocysteine was low in this population. And they opened the whole methylation cycle with methyl donors. So with B12 and folate and betaine. They normalized homocysteine levels. They actually increased homocysteine because homocysteine was low, but they also dropped, the elevated methionine and glutathione synthesis. So glutathione, being produced from the sulfur in homocysteine, improved. It was really cool. So by addressing the methylation cycle, it just had these far-reaching effects.
Yeah, and didn't she have like sibling control in that study?
I think she did.
Yeah, and then one of the siblings had Down syndrome. So she found a similar methylation impairment in Down syndrome.
Wow, that's fascinating, which has been identified elsewhere. It's been published on elsewhere. That's very interesting. Okay, so that higher methionine, we're thinking about, maybe protein consumption, animal protein consumption, but perhaps more likely just methylation.
Yeah, looking at those things, because so you have the methylation cycle and then goes to cystathionine, and then cystathionine goes down to that transsulfation pathway. So, what we're looking at there is cysteine, which, plasma cysteine can be rate limiting for glutathione. So,
and how was mine? What was my cysteine?
Yours was on the lower end.
Yeah.
Yeah. So that's some good research. I think there's a whole blog on that on your site. So that low cysteine, which when I saw the article on that, I actually contacted the author from the Netherlands, and I'm like, didn't you mean cysteine? And he's like, nope, I meant cysteine. So that's on the lower end, and you don't want it super high either, but it's going to help that feed into there. And
but so earlier, the reason cysteine is important, everybody is aware of cysteine, homocysteine, cysteine, and glutathione, but cysteine is actually 2 cysteines complexed, and that is how it gets intracellular. So that's basically how glutathione is produced.
Yeah, so it's going to come in, and then you're going to have like the glutamic acid's going to come out, and cysteine's going to come in. It's going to feed the cysteine. So, and that's going to go get together with, glutamic acid and glycine.
And make glutathione intracellularly.
Yeah, so you have a low cysteine, you have a higher pyroglutamic acid. So do you maybe need some support there?
some detox support. And I, when we were talking about this, so actually, so we could jump into that. And then I had a really high, what was really high that
It
we
was
puzzled over for a minute.
Yeah, so we look at some toxic markers. We look at mandelic acid and benzoyl form. We also looked at different xylene markers, 2, 3, and 4. But so your mandelic acid was on the high side. It was over the 95th reference.
It was high, frankly, high. Yeah. Yeah.
So, that seemed a little bit puzzling because I was say only once have I seen somebody with less than DL on all of them. Most people just get exposure to, mandelic acid's going to come, just from tar production, from petroleum byproducts that they make things. You can get car exhaust, you can get contamination in food packaging. But so yours was a little bit higher, and then you realized that you have been getting.
Yeah. So it turns out that we have had, just massive construction road work going on just outside of our house it's just wrapping up now. It's been going on for, over a year. There has been a ton so recently paving is the last thing that they do. Recently, they did all of the paving, including a chunk of my driveway. They expanded, they put sidewalks in. It was a massive project. And it seems obvious, right? Like, of course, that's your exposure. But because it's been just in our lives for this chunk of time, it wasn't top of mind. But of course, I've got evidence of exposure to petroleum compounds. I mean, tar has just been in the air. It's interesting, even though we've got, air purifiers in the house, and I keep the windows, close to the construction closed. Of course, I've got some of that background exposure. I mean, I need to do this on my daughter as well, and suggest it to my neighbors. There's 3 of us, 3 houses, like sort of together in kind of the heart of what's been this project, and it's thankfully coming to a close, and it has made everything better there. But yeah, I've been under this lasting toxic exposure that you've picked up exquisitely, and we can see that it's influenced my my glutathione. And I when I first looked at these labs, I was like, what,
that can't be me.
Yeah, where am I getting this? And as you and I just sort of teased apart the pathway, and I thought about it, I'm like, oh my God, it's huge. And this is, I don't know, I wouldn't, this is not information that I was getting anywhere else with this level of clarity, this level of sort of granular detail. What I need to do, what's happening, what the exposure source is, how it's influencing my detox pathway, but also how it's influencing, negatively the aging journey. I mean, you can see how this is where it starts, and if I continued to have
bit
these
by
exposures,
bit.
Yeah, it's true. I know it's true. We're like going back to the Peter Attia idea of, medicine 2.0. This is medicine 3.0, where I get to correct this.
Right. And I will say, if you want to test your daughter, this was the urine part. So you can do just the organic acids. So you don't have to do the blood draw.
I absolutely want to test her, and my intervention is going to be now, like, in your opinion, what is the best approach? Do I use, preformed glutathione? Am I using glutathione or NAC plus some,
I mean, there's not one great study that has done a comparison, which reminds me, I got the lottery, the big lotto's up to $1 billion. And that's what I always say, like, I'm going to enter that lottery and I'm going to win $1 billion, and I'm going to do so much functional medicine research. I'm just going to answer all these questions that I have.
And you will. I love it.
But yeah, so I think that I would think about glycine and NAC, because you also, I think, had an slightly elevated benzoic acid. So, which,
same. It's probably from this same exposure that's been happening.
Right. So you can get it, we look at it in the microbe section, microbial metabolites. You can get it from diet, you can get it from microbes, but you can, get it other places. But it should conjugate with glycine and become hippuric acid.
Right.
And yours is,
Oh, but my hip here, I'm not making that conversion.
yeah, so you're not doing that. So is there
Is
not
that
enough?
because I'm stealing my glycine for its toxic burden? Is that what you're thinking?
I'm not even sure why you're doing that. I think there's other things. Glycine does a lot of conjugation stuff, but just looking at that, so your benzoic acid isn't over the 95th, it's at the 90th. But most people are less than DL because it just gets cleared out all the time.
Right.
So, you know, I think that just another reason that I would think of glycine. And then it should go over to your hippuric acid. So generally you'll see hippuric acid higher and benzoic acid all the way low. And so
Yeah.
I'm not seeing that in you. Elevated hippuric acid's been associated with a, you know, more Mediterranean style diet. It's been associated with better gut diversity. So looking at that. But I was going to say with that, that mandelic acid, you also had glucaric acid, which was elevated.
My Mediterranean diet. I'm noshing on this all day. Actually ran out of salad dressing.
Finish it up. Yeah. Yeah. I mean, and the mandelic acid, the benzoyl form, those have been like collected by NHANES data for a long time. So you can actually break it down, you know, NHANES, where they look at 5,000 Americans, I think it's every 5 years or 10 years, and they just look at everything. So you can break it down by gender, by age, by location, if you're interested in all that. But I think that that was, you know, one of the things that I would think of with your detox. So thinking about that you have an elevated pyroglutamic acid, you have a you know, what looks like some need for, which can identify glycine need. And then you have that benzoic acid that's elevated, so more on glycine. The cystine that's a little bit lower, the somewhat toxic exposure. I think those are the things that I think stood out.
Okay.
On your otherwise really good looking report.
You've inferred it's more glycine because you're pulling in a handful of other analytes. So pyroglutamic acid could indicate cysteine as well.
Right, and your cystine was low.
Okay.
Well, your
So I do
cystine was
need some
low.
cystine
Yeah,
support.
so I would
but
do both of them.
We can also clearly see glycine support. So those are two of the three amino acids, and I need both of them. Glutamic acid, do I need glutamic? You looked at my glutamic. Wasn't that a little bit on the lower side?
I think your glutamic acid was actually, yeah, it was on the lower side.
So I need all three aminos. I need, so preform glutathione wouldn't be a bad idea.
It wouldn't be a bad
plus
idea.
Another kick of glycine.
Yeah, and there's some research, like if in research that specifically, and I'm not putting you in this category, specifically looks at frail older people, that glutamic acid tends to become elevated or becomes low in people who are frail.
Interesting. Glycine is a longevity amino acid. I know it's sort of, moving up the ranks of getting attention these days. So we want to have enough of it. And
yeah, I mean, it's just like, so I think there's some really good long-term research when you look at metabolomics and metabolic concerns. So these are the research that they've looked at, the Framingham study, the Primed study, that they've looked like for a long time, decades, and they've associated higher risk of metabolic issues, diabetes, obesity, in people who have elevated aromatic amino acids. That's going to be phenylalanine, tyrosine, the branch chain amino acids, higher glutamic acid, and higher lactic acid, higher alanine, those, and then lower glutamine and lower glycine.
So that pattern. Wow.
Yeah. And so I did look at, that's another one, that branch chain amino acids can become lower as you age. They're, and the assumption is that they become lower because as people get older, they have less muscle mass. So it might be related to sarcopenia. Some research has noticed that it starts to decline when, people are in their 30s. There's a really interesting article, and they put people in categories of, 20 to 30, 30 to 40, 40 to 50, 50 to 60, 60 plus. And that they looked at branch chain amino acids, and they looked at cardiorespiratory fitness, and then some other things, and they have it in a real nice, I mean, if you've got a nice pathway or a nice diagram, I'm all for it. And really everybody was maintaining it until they got to 60. And at 60, it just kind of dropped. The branched chain amino acids dropped and the cardiorespiratory fitness went up. So, it can have a big drop. So when I'm looking at
cardiorespiratory fitness,
Mhm.
their fitness increased or declined?
Decreased. Their cardio fitness declined, and their branched chain amino acids declined.
Also declined.
So they're, you're thinking of it. I'm going to look at, I have your report like on another computer.
I do, by the way, I just got my VO2 max done
and it
recently.
Was excellent.
I was in the 90th percentile for my age. I went to, at UConn, I'm here in Connecticut. There's the Corey Stringer Institute, excuse me, where it's an incredible place. It was so fun. It was really fun to hang out with the PhD students and get tested. And it was exciting. I brought my bike up there. We hooked it up and I did my lactate threshold. They took a bunch of blood specimen, and it was neat to see that even, with this busy world that I live in, I do put attention into exercise. I put attention into muscle mass. I put attention into cardiovascular fitness, and I'm, doing pretty good. So,
yeah, I mean, I think exercise, I know you're a big proponent of that. One of the other things that, to mention with exercise is it can clear kynurenine. So that's what was one of the other things, like your kynurenine was low, but maybe the exercise, kind of helped bring that down.
Interesting. Yeah. And we saw that my lactate was high, and I said to you that it's certainly possible that I had done a more intense event before I gave the specimen, and I perhaps should have controlled it. So the day before I might have, done a more intense resistance training or high intensity intervals on my bike or something like that.
I think just knowing what you've done before is, otherwise you don't need to make it perfect for the test.
But if your lactate is up, if you're in anaerobic metabolism and you're sedentary, that doesn't bode well. That's a strongly pro-aging pattern.
Right. Yeah, I mean, and there's some good population studies looking at that. I think there's a big Dutch civil service study, and they just looked at urine lactic acid and alanine. They looked at, they found a lot of things, but those were two that really stood out with the whole kind of glycolysis process, that when those are elevated at baseline 5 years later, those people are more likely to have metabolic issues.
But it makes sense. I mean, it just makes sense.
Yeah, I mean, following the pathway, follow the pathways, just pull them up and you can kind of see where they're going. With your branched chain amino acids, they were fine. They were, a little, valine was a little bit higher, but overall they were great. Your alpha keto acids, so looking at them all together. So I don't want them too high because that's going to be associated with metabolic conditions, but I don't want them too low because that's going to be associated with,
with aging,
sarcopenia.
So you just want the sweet spot.
Yeah.
This has been great. I mean, I can see the areas that I need to work on. I've got a really clear action plan. I know why I need to work on these areas based on, exposures. I'm grateful that the construction is done. We're not going to have any more tar being dropped in our neighborhood. I certainly hope, so I can start doing this cleanup. I want to evaluate my daughter, and I'll talk to my neighbors about it, see if they're interested, because we'll all have this.
Yeah.
We'll all have it. What else, Betsy? Like, what else on this report do you want to bring out? We talked about toxins. We talked about some of the really cool patterns associated with aging and what we might do about it. There's a lot here. And in fact, I would just suggest, and then I'll shut up, it's for anybody diving into metabolomics. It's fun, it's powerful, there's a lot of action, and you could certainly start by using the ready reference page one, but also consulting. You're available. People can
Yeah,
talk
we're
to you
available. And,
yeah,
we have stuff on our website. You don't
yeah.
have to even sign up on our website. You can just go to the Learning Center. I have a webinar that's on aging and metabolomics. That's
Awesome.
on the metabolic function of metabolomic. That's on mood disorder and metabolomics. There's one that correlates gut function and metabolomics because that's so closely put together.
And you and I have a previous conversation when you were first launching the panel. So I think it's going on 2 years now, that we'll link on the show notes as well. So we talked about some pattern analysis there. All right. So just in the final minutes, what else? Tell me what else we want to talk about.
Well, the other one that I think that I briefly mentioned was alpha ketoglutarate, alpha ketogluteric acid. And that one has, it's been around a long time. But people don't always pay attention to it. And yours is good. Yeah, higher. Like certainly they have supplementation with alpha, AKG, and that's been shown to be successful. But
well, it's actually been shown to lower biological age as by 1 clock measurement, and it's also known to drop with age. So
right. Well, from 40 to 80, there's a 10-fold drop.
Wow. So it really drops.
Yeah, right. I know. I'm smack in the middle of that. So it's like, okay.
And it's sort of the heart and soul of the Krebs cycle. So it's right in the mitochondria. It's a key player in generating energy. It also happens to be a cofactor in demethylation on DNA. So we need it to take methyl groups off of the DNA. We need alpha ketoglutarate to be able to do that. So if you're hypermethylated, and you don't, and if you have a good gene turned off with excess methylation on it, then, you need AKG to do that cleanup. But it plays some very important roles in the body.
Yeah. And with more acidic situation, it can go down. So,
Oh, so that would be like inflammation?
Yeah. Or people who maybe shouldn't, I think of some of the research they've done, like people who are getting a more plant-based diet. And I don't think they're necessarily looking at vegans, but just making sure you're getting all those plants, that was associated with a better level.
Interesting. And why? Because they were more alkaline? I mean,
I think when it's more acidic, it gets absorbed. So they have, that it under, more acidic conditions, it gets absorbed and results in a drop in just urinary excretion.
Oh, okay. Huh.
So we're looking at urinary. I mean, I think that the Krebs cycle, it's so fascinating. I think, years ago it was like, well, this is high, so let's do that because, there are a lot of nutrient cofactors there, but everybody flows into there. Fats flow in, glycolysis comes in, all the amino acids flow in there. It's connected with the urea cycle. So it's just like everything's happening there.
In the mitochondria, in the Krebs cycle.
Right.
Yeah.
So
cellular respiration. I mean, it's metabolism, right? It's sort of like
right. It is
the soul of metabolism. Cool. Yeah. So of course everything would go there. Protein, fat, carbs, all ends up there.
Yeah. I mean, and I think as I guess thinking of last words, some of the things that, in researching all this and getting it started and looking at, researchers are putting it out there, that it may be, it's going to become one of those things that you just do every year. Like you're going to look at certain markers every year. And this is really going to tell you, and it's good to compare against yourself. Yeah. Because there aren't necessarily standards that, there's healthy reference range. Yeah, we have that. But for you yourself, like what pathways are impaired and, so oh, that's a change, so you can address it differently, but just getting it every year. I mean,
well, especially, as we we've been talking about aging a lot. We're both interested as we
We're
march.
both agers.
We're both agers. I mean, there's a lot. I want to make sure that I'm as dialed in as I can be here. Just like I took the time to do my VO2 max and, take the time to exercise and, work on living, eating well, etc. Like I want to know these are dialed in.
Yeah, this is just going to be at a much more a smaller level. Yeah, you can see I got good muscle tone. How's it all functioning?
Yeah. One of the things we were going to talk about, and we forgot to, so I want to bring it up because I think it's really, really cool, is you look at equol. And so let's talk a little bit about equol, because it's an awesome compound in my opinion. It's an essential, important player. So what is it?
So we have our section 6, which is microbial metabolites. So it's a combination, just the markers on the page, that's where indole acetic acid is listed too. Like it's looking at what bacteria do you have in your gut? What are they getting? Like what's coming into the gut and what those bacteria are making. So looking at all those metabolites. So you had good indole acetic acid. You have some polyphenols there, but one that you had was the equol. So that is going to be, it gets made and it's got certain health benefits. And it's made from isoflavone. So certainly everybody's, thinking of soy, but it turns out the nut that's highest in that is pistachios, and you're a big pistachio eater.
we always have a ton of them in the house. Like it's just sort of a background item in our lives. Pistachios. I mean, I probably have a handful of pistachios, maybe 2, most days. Yes, I'm so, for me in my research, we're looking at so in the aging process, you start to hypermethylate and turn off incredibly important genes that keep us healthy. So another angle of looking at aging being the biggest driver of disease are these hypermethylation patterns. So we start shutting down protective genes and turning on pro-inflammatory genes. And one of the things there's a lot of research looking at the tumor suppressor genes getting hypermethylated as we age. It's like we're just shooing in cancer. It's crazy. And equol, so there's a whole table in the book that I authored, Younger You, and looking at these tumor suppressor genes, and equol is a key player in supporting the re-expression of these genes. And I want to say that most of this research is done in vitro. There are some animal studies, actually sulforaphane is badass for helping turn back on these tumor suppressor genes and being effective in animal models. But in our research in humans, we can see now, and this will be published in the not so distant future, that we've had a favorable influence in our diet and lifestyle intervention on tumor suppressor genes as well. So it's incredibly exciting. But equol just shows up over and over and over again, equol. But we need to be able to make it. We need to be able to make it in our GI. So I was absolutely thrilled. And I know a lot of people are anxious about eating soy. I'm not, if we eat good quality soy and we can tolerate it, if it's organic. But pistachios. I mean, I don't eat a lot of soy. I do eat, every once in a while I will, but it's not a big player in our lives. So as you and I were brainstorming, like, why do I have equol? I had to look it up in my own book. Oh, pistachios. Wow. I didn't realize that.
Number 1 nut. So be cracking.
I'm absolutely thrilled that I've got background equol.
It's a nice little thing to have. I think when they look at population studies, probably 25% of Western countries
have the correct. Wow.
Yeah.
You have to have some of that. It's nice. So it may be a little bit of a surrogate suggestion that I've got some diversity happening with my microbiome, that I'm able to make it. I think people in Asia, soy consuming countries, have way more equol.
They have,
and this is associated with lower risk of breast cancer, less transition to menopause, just less symptomology with menopause. I mean, equol is just a really important player. I mean,
Yeah,
I'm a fan.
it's a nice little bonus to have.
It's a nice little bonus. I was pretty psyched. I was really excited to see that you were measuring it because I was just waiting for a lab to make it available to us. But then B, yeah, I didn't anticipate being a producer, so I was pretty happy to see that. Dr. Redmond, it was just great. This was a really fun conversation.
Yeah, I like confirming that your health is good. You're going to make it another year. Yeah,
I'm making it, but I've got my marching orders. Thanks for joining me. We're going to gather all of your papers. I will be downloading some of those papers myself. We'll get a sample report so people can walk along with us as we take this journey. And I look forward to seeing you in the not so distant future and having another conversation on new frontiers.
Yeah. Would like it. Yeah. We'll do a follow-up.
We'll do a follow-up.